Hey guys, can you hear me all right? Nice. Can you speak just so I can make sure I can hear you?
Hello, hello.
Hello.
Testing.
Awesome.
Hello.
All right. Hello, Aaron. What's
Yo.
going on? Good to see you.
Good, man. How are you?
I'm good. I'm good. Long time.
I know, right?
We are
well?
live, gentlemen. I think we have Lucas join us too, and Peter as well from CerebrumDAO and the Gale team. Let's let people stream in here. I'm going to share my screen too. Okay. And Brian also from the Gale and Cerebrum team. Hey guys, how's it going?
Hey, great. How's everybody?
I'm good. I'm going to share my screen because literally, I think we just had the token go live. I think just in the past few minutes. Ada, if you're listening, if you want to push the announcement too, that'd be great. Can everyone see my screen? Yeah. Cool. All right, so we are live. The Gale token has just gone live on the markets after a pretty amazing sale given the circumstances. So I'm going to stop sharing real quick and go back to the screen. Before diving in, just some background for everyone tuning in. The Neuron Gale ignition sale has just wrapped up after 24 hours live. I think we had about 14 plus oversubscription on the sale. I think I saw last over 700K in USDC committed. Given the markets and the meltdown in the past few days, this is pretty amazing for me. So I'm very psyched to have everyone on. So maybe let's just go around real quick and do a round of intros. So yeah, why don't we start with Peter from the Gale team? Can you guys hear Peter? I can't hear you.
I can't. No, you might be muted, Peter.
Doesn't look muted. Oh, no, still nothing, Peter. Do you want to try to rejoin, perhaps? That might fix things.
Let me try this. Does this work?
Yeah. Yes.
Good. So many microphones, so many options.
No worries.
Okay, good. Hello, my name is Peter. I'm part of the Cerebrum team about a year now. I am currently the head of translational science, as it nicely says, and I've been involved in the discussions around Gale for, as we had, first encountered this fantastic opportunity. So my background is I'm a molecular geneticist, and I've worked in pharma for about 25 years in drug discovery and drug development. So that's my interest in the new things that can happen.
Great to have you here, Peter. Brian, do you want to go next?
Yeah, absolutely. So hi, everybody. I might be a familiar face for some of you. I'm Brian Mierski, founder of CerebrumDAO, and lead the overall strategy and direction of our network organization. Super excited about this. Really tough markets, I think, for those of us waking up in Europe today and probably in the US now. It got worse overnight, but not for DeSci. So super excited about this. The Bio Launchpad. This is our first token sale on this new version of the Launchpad. And for those of you unfamiliar with the Cerebrum model, Peter is part of our team. He's an expert in translational science, so translating research into drug candidates with deep expertise. We've got a pretty strong bench of neuroscientists, but also people with drug development expertise on the team and advisory. And we've got a great deal sourcing engine. So we're looking at so many tremendous opportunities that we think can translate into novel therapeutics in this field of neurodegeneration broadly, Alzheimer's specifically, which is just a massively unmet need. So the more successful this launchpad is, the more of these types of projects Cerebrum can bring. And we've got a few exciting ones in the pipeline already. So really thrilled to see the support from everybody out there. I think we were almost 2,000 participants, $700,000 for what was a modest $50,000 starting raise. So even in these turbulent markets, you guys showed up and are funding some important things. And we're here to go make that happen and bring it to reality.
Awesome. Let's do some intros on the bio and molecule side real quick as well. Lucas, do you want to intro yourself?
Yes, happy to. Can you hear me? Yeah, should work fine, right? Yeah, nice. Yes, Lucas here, working on the bio side of things as an operations manager, but also on the science side. I have a background in medicine, so I'm a medical doctor, but also very interested in translational biotech. So I'm very excited that the project was able to fill so quickly once and kind of show all of this traction, and there's still interest in detail. So I'm very excited to be here.
Cool. Aaron.
Aaron Weaver. I'm the Chief Commercial Officer at Molecule. Spent a life in finance, also as a biotechnology VC. Spent quite a bit of time around CNS disorders also. So, what I find really exciting about the project here and what's happened in the last 24 hours is the ability to get more research into novel mechanisms, and particularly within the area of neuroinflammation, which is becoming very, very topical in terms of neurodegenerative research.
And lastly, Rafa.
Hi everyone, my name is Rafael. I work as scientific assistant at Molecule, also supporting here the bio team. My background is in biochemistry, synthetic biology. Very excited about this project because I think it cannot just be used in Alzheimer's disease, but the broadly central nervous system space. So this has huge implications here. I'm very excited. This is super, super, super important work.
All right, so I want to keep things pretty casual. I think let's kick off first. Just anyone have any sort of first impressions of Brian? You mentioned earlier, obviously the markets are in a tailspin, the broader markets, but we saw a lot of people show up for this sale. It smashed through its initial funding goal. So any sort of initial reactions to how the sale went, and especially in the sort of broader context of the macro shifts we're seeing in crypto?
No, it was smooth and fantastic. So it is exciting to see, especially with the new mechanism now, and people are coming and committing US dollars. So people are putting into USDC, and to see $700,000 pledged for this project gives me a lot of hope and excitement about where we are, even in today's market conditions, that people want to see this science funded. And we're having these discussions a lot now, and I'm talking with a number of different researchers as well, getting them really to understand that we're funding into this translation gap, this valley of death, and there are so many promising opportunities out there for novel therapeutic candidates. They just lack funding, and it's been getting worse with some of the reductions in government funding, at least in the US. So to see that, people in this market in VSci are stepping up and trusting an organization like Cerebrum to find those projects and to shepherd those to opportunities is really great. So I'm hopeful that this is a harbinger for the next set of projects. If there's 700,000 in demand already, and this one gets off to a successful start, like it looks like it will, that even more demand comes for the next wave of projects, and we can fund more great science.
Yeah, I don't come from a medical background. I come actually for the past 7 years been in crypto, and I wonder every cycle, every like major shift, whether we're finally going to sort of expand beyond the degen casino that is, the vast majority of crypto, and whether, we see so much, we're in this bubble of crypto, and it rarely extends beyond, X in the crypto sphere, and getting sort of real customers, real people, and Web2 generally involved. And so I wonder if sort of like a silver lining for these markets is that, a lot of the energy is going to start to shift to bringing sort of real economic activity, real, tokenized IP science on chain, and sort of measuring projects also by revenue too, that this tokenized IP can create through, let's say, licensing or products and all that. So I think there is potentially a silver lining to these markets, and I think this is an example of maybe a shift happening.
Well, I hope so, and because we've talked about this for a while, and there's a whole category of real world assets that gets a lot of attention in crypto, and they always talk about tokenizing things that kind of exist in financial markets already. And I've always thought, and many times when I explain this use case of IP NFTs and IP tokens and tokenizing intellectual property and making markets in the early stages of this, I get a reaction from people who are generally skeptical of crypto that say, wow, that's one of the first use cases that actually makes sense to me. And when you think about real world assets and on chain, and we're now in a digital financial system here with blockchains, creating a new asset class of intangible intellectual property and tokenizing it and making markets in it makes the most sense to be the first real breakout use case for real world assets on chain. So I think it's almost inevitable that IP will be the first breakout real world asset case on chain, just because of that. It's an intangible asset, and it's never really existed in a financial market sense. And we can make that happen in the digital economy on blockchains. I think it's been years in the making, obviously, and we've been putting in the work along with Bio and Molecule and the other Bio DAOs for years now. And these things don't happen overnight, but hopefully we are pretty near that first inflection point where we get some broader recognition for this asset class that we're creating that has, tangible value behind it. And you're right, we are going to see economics come out of this, because this stuff can be licensed into industry that will drive royalty streams and revenue, and it will actually be products that people will use. In our cases, hopefully therapeutics and drugs that are preventing and curing diseases.
I'll probably add something around that. I think industry has also been screaming out for a market mechanism that helps bring forward some of these novel mechanisms, and through a matter of validation where they can then pick them up, where it sort of sits within their institutional mandates. The case for neurodegenerative diseases is like pretty well known. We have an aging population. It's a shared issue globally. You look at how capital allocation worked previously, it was concentrated on a very select number of cities, pretty much within the United States, and maybe, in some elements of Europe. But this is such a profound issue, and a lot of people are personally touched by it. I am, with members of my family. Now I look across my my group of friends and all that. We have aging parents, etc., who are now really starting to suffer from this, and just looking at the absence of solutions, and just also how the direction that R&D has traveled in over the last few years, which has been very consensus driven and non-exploratory in a number of ways. And so, there's a periphery of advancement in certain types of biomarkers and ability to effectively assess the underlying cognitive states of a number of the patients. And so this is really a critically needed thing for the advancement of human welfare.
Well, you touch on an interesting point, Aaron, too, as it relates to neurodegeneration. And yes, the macro trends with the aging population for sure. And environmental factors are also starting to play a role too, as we see, microplastics being pervasive. I see it with the biomarkers now. We are on the cusp of surrogate endpoints and predictive biomarkers for a condition that we really haven't been allowed to treat because the diagnosis happens so late or after death. Yet the pathology we know develops for decades before. And so the ability to get in those early decades and actually know that this is happening and to slow it down or reverse it, is almost here, which is absolutely the perfect time to be funding these new strategies that are looking at neurodegeneration and various diseases like Alzheimer's as a function of aging and trying to go upstream, which has been exactly Cerebrum's strategy. And I really think we're going to have pathways to get drugs to market like we have been in other disease categories because we can get into the surrogate and predictive endpoints finally. And I think that leads a lot of promise for the pipeline that Cerebrum's building to be potentially successful.
One other point I'd probably add around this, and this is also why I find a moment like this really, really significant. In my days within TradSci, we've tried to support endeavors or research endeavors within the neurodegenerative space where these are novel mechanisms. We're exploring different types of brain biology, et cetera, to sort of have rest of industry sort of say, hey, we want you alone to carry the load on effectively proving this. And, what we felt we really missed was a community of people to help sort of like pull that all through. There is a point that, industry will engage. And if you say, for example, you take a leaf out of Peter Attia's book, he talks about the 4 horsemen of disease. And it's like metabolic diseases, cancer, like cardiac diseases, and then effectively neurodegenerative diseases. 3 of those get a lot of funding out of pharma, and they have existing treatment pathways. But we know that people probably suffer the most in terms of neurodegenerative diseases because it is just a loss of faculties over time, and it bears such a significant emotional toll over people. And this is a shared burden for people effectively across the planet. And so for something like this to be able to like overcome the cold start problem that a lot of things within TradSci effectively experience, I feel is a very meaningful moment, hopefully the sign of more things to come.
So maybe we can dive specifically into the science behind Neuron Gale and why this specific research is so timely, important, and the sort of promise if it's found to be effective. So maybe if Peter or Brian, if either of you want to take that.
I can make a start. I can kind of give a rough explanation of why it's very timely and why this is very interesting research. Just to start, lectin-3 has been studied for more than a few decades now and has been specifically looked at in a number of systemic disorders, cancer, fibrosis, inflammatory disorders, and such. It plays an important role in immunomodulation, immune regulation. And it also in the space of neurodegeneration, it has recently come up as being an important player. So it was first coined as a biomarker because it was clearly associated with a lot of disease activities. And given the function of lectin-3, that makes sense. So it has been also coined, as I said, as a biomarker, but then people started to look at the moieties of lectin-3 and realized actually that it was targetable with small molecules. And that, of course, in the current space of developing medicine still is a very important space. Small molecules have, of course, always been very important in treating disease, but they're very convenient. You can put them in a pill and you can take them in certain drug regimens, as we call them, as opposed to antibodies, which can be very effective in certain diseases like cancer, but obviously require a completely different treatment setting, and beyond that are often, of course, very costly. So galectin-3 is as a target, as I said, has been already recognized. It's a very competitive space. If you look up galectin-3 drug discovery and drug development activities and look at the patent space, it's quite dense, specifically in areas like cancer, inflammatory disorders, and fibrosis. There's no approved molecule yet, so it gives lots of perspectives. That's why a lot of companies are very active on it. With regard to neurodegenerative diseases, that's a lot different for various reasons. You already pointed to it. Neurodegeneration, or CNS as a whole, has been an area probably with one of the biggest attritions in this space of drug discovery and drug development for various reasons. Not one of the least is because the brain is so inaccessible, both from understanding how a disease is as well as treating it, because the endpoints often cannot be a biomarker. So you have to focus on very subjective endpoints. So that's the reason why a lot of people have been holding back, also probably with galectin-3, because it's much more interesting as a company to develop something in cancer as it is in neurodegeneration when you're looking at the future space of likelihood that you can commercialize it. Not so much for non-met medical need. I think if you compare it from a non-met medical need, there is clearly a need to do it in diseases like Alzheimer's disease. And that's where we stumbled across galectin-3 with quite some good, I would say, not just descriptive data, but real evidence that there is a direct link between galectin-3, neuroinflammation, and Alzheimer's disease as such. And again, here, galectin-3 can be considered as a more universal target, not just for Alzheimer's disease, but also for other indications. So, and it's a targetable molecule. It has pockets which you can fill with a small molecule. So our traditional way of approaching this is, I would say, fairly easy. You identify a pocket, you do your medicinal chemistry. You can even now, under the current circumstances, design molecules, because also the structure of galectin-3 is well known. So you don't have to go to the traditional ways of searching for molecules. You can actually design molecules. So the whole space is actually set out for us now with all the knowledge that's already there, right? As I told you, there are decades of understanding of galectin-3 and quite some drug discovery and development activities for systemic diseases. The challenge for us will be, to move into the space that we can develop a small molecule that can actually pass the blood-brain barrier and have an effect on galectin-3 in the brain. And that is the next step. The first step, and I think this is an exciting step, is actually to prove if we intervene with galectin-3 and neuroinflammation that we actually can see an effect. And that's the first step we would be taking now. I think this is also, again, I was explaining this morning also in a meeting with Brian, the interesting space where we are. You can actually do this whole discovery process in smaller steps, rather than considering this as a huge project where you would need to have an investment in. Now you can take small steps to further when you actually start to embark on the big mission to develop a molecule. So I'm quite excited. I think it has lots of opportunities. I'm also very happy to see that we're not the only ones seeing these opportunities. So yeah, Brian, maybe you want to add something to this.
No, I think that was great, Peter. And you're right. Like we're finding a lot of success and really, thinking outside the box. I think this new model of funding through tokenization and through launch pads is really forcing us to get very creative on what are tangible early milestones, and just kind of build almost like a rolling thunder on these projects of like let's do some really early proof points and start building confidence at an increasing level, so we can attract more and more capital as we move it closer to preclinical and pre-IND status. So I think that's been great, and we found some real innovations as we've gone through with this. We've been careful on some projects to make sure we do replication studies. Like this project, we're verifying that the mechanism is going to work before we go into a little bit more expensive. There's really strong reason to believe it will. We've seen even with our Percepta project, we've really figured out how to do a bona fide, full-size, decentralized clinical trial with 154 participants on a double-blinded, placebo-controlled basis with at-home blood-based biomarkers that are at the earlier stage of detecting things like amyloid and tau pathology. So at a fraction of the cost. So I think we're moving faster, and we're doing it less expensively. And I think that shows a lot of promise for how we can build what will be a high confidence pipeline of opportunities in this massive space of neurodegeneration. So a lot of green shoots, but this is a great addition to the portfolio for us, and really eager to get underway and get some early data relatively quickly to the community, so we can continue to build confidence on this particular project and target.
Yeah, you mentioned this, how this sort of model is using these smaller pockets of funding to kind of de-risk the science in phases. And I think the R&D is kind of mirroring the evolution of the financial models too, or at least the fundraising models, because this is actually the first time that bio is releasing its sort of milestone-based funding through season 2 of the bio platform, whereas the kind of traction grows, as you guys, your token hits these different valuation levels, it unlocks more funding for the project. That's as opposed to these bigger, chunks of raises that we were experimenting with, and that biotech is all too familiar with doing that up front. We did this sort of smaller 50K raise, and then in the market, as the valuation grows, it unlocks more funding all the way up to upwards of $1 million plus. So super excited to see this kind of experimented with in real time, and allowing these sort of smaller chunks of funding to respond to different developments in the science as you guys unlock those new data packets and all that.
Yeah, it fits the operating model that Cerebrum has had for some time now, actually. So we've been treating our projects this way. Another project that Peter's been leading too is our Fission Bio project around mitochondrial dysfunction. And that was one where we did have a large fundraise. Cerebrum committed a fair amount to an early milestone. And then we went and raised another large chunk of capital. I think all told, it was probably well over $400,000 dedicated to that project. To date, we've spent, I believe, less than $100,000 on that. And we're through our second milestone on it, which is ahead of progress from even the first tranche of funding. So we've been very careful at producing results, thinking creatively about how to get there, and trying to be a lot more cost efficient, and reporting to the community to build confidence in these projects. So this is we're kind of used to this, I guess, in some ways, because that's kind of the model we set up. So we're excited because that means, to the degree that the Launchpad continues to perform at this level, we do feel like we are capable of feeding it some really interesting opportunities for the community.
I guess, Aaron, I'd want to know, how pervasive do you see this is, that sort of biotech in some ways is potentially adopting this sort of lean startup, sort of rapid, R&D cycle model? Do you see that across the industry, or is it just kind of starting to bubble up in DeSci?
It's getting there, I think, across traditional venture-backed entities, but it really is a matter of necessity for a lot of biotechnology companies, because we've just had I think this is also sort of representative of the broader venture ecosystem, is we've had a lot of monetary deleveraging, which has meant that risk dollars have sort of come down in terms of lots of different venture asset classes. But it certainly seems like it is the best aligned approach to be taken for markets, because investors want to see the tangible progress before any further money unlocks, and they really understand the scientific progression and validation of these assets. And as we know, I mean, this is an R&D capital-intensive industry, and you want to make sure that money is wisely spent along the way and that milestones are effectively being hit. I just think that in terms of technology tooling, DeSci just is able to offer a lot more, because it's like it's not paper-based obligations and then money just sits in a bank account and then people can draw on it in violation of those contracts. And I mean, that certainly happened a lot in traditional industry, whereas here, there is complete transparency around how this effectively occurs, and and then like Coda's laws, so like smart contract embedded mechanisms that actually then permit the further release of capital.
So maybe we can dive into the specific roadmap that Neuron Gale is plotted out. What are these sort of development milestones that are trying to be hit, which potentially we could see the sort of market react to and this funding unlock. So I know there was a roadmap shared in the announcement, but maybe to just go over like what specifically does the team plan to do over the next, let's say, coming months. Maybe Peter or Brian, you can take that.
I can take this from the start, and Brian can add. The first steps are fairly easy, as I said in the beginning. In principle, now we want to make sure that we understand, next to descriptive, but galectin-3 is important in the pathophysiology, that actually by inhibiting it, you see an effect in the main biomarkers in neuroinflammation. So that's what we're going to prove in a 4-week study with a mouse model that we think is relevant for AD pathology. We can argue about how relevant these models are, but at least from a biomarker perspective, they will tell us actually if there's a direct relationship by inhibiting the molecule and seeing a number of biomarkers change. And that's what we're going to do in a 4-week, relatively simple experiment. Galectin-3 inhibitors can be bought just by any supplier in a high purity. So these tools are there for a good cost, and we're going to infuse them directly into the brain with a pump system. And this will allow us, by looking at brain pathology in a mouse model and at the same time at a number of key biomarkers in inflammation and AD pathology, tell us if there's an effect. Now, the end will probably never be black and white, will probably always be gray, but it will help us to take decisions. What's going on here? Is the specificity that we're looking for good enough? Is there other aspects maybe that we didn't completely understand with regard to the pathophysiology of AD and specifically galectin-3? But it will be greatly revealing and a huge important de-risking step. Because again, if it would be black and white, and it would really be, we don't see anything, and we are sure about, that all the experiment and all the aspects have been traced, this must work. If it doesn't work, the concept doesn't work. It's a nice biomarker, probably correlates, but there's no causation. And then we can bury it. But I'm quite confident also, as I said before, it's not just neurodegeneration that this has been tried in. It's a lot of other diseases which already are in phase 2 or phase 3. So the causality is there. If it's in the brain, that's what we're going to prove. And then the next step is going to move, of course, to a more relevant way. That is to actually look for a molecule that we can administer in a normal way and look over a longer period of time. The experimental setup we chose now is technically limited. We're pumping the drug into the brain for 4 weeks. That you cannot do over periods of months. And looking at treating these mice, we have to look at over a longer time, because also aging is an important aspect in these mice. And of course, prolonged treatment is an important aspect. So the milestones are really almost like a traditional drug discovery project with, I wouldn't say it's an exception, but with clearly making sure that the first step is being de-risked. You're mute, Nate.
Sorry about that. Sure. And Brian, anything to add there on the, what you're most excited about the roadmap coming up, and maybe, also for folks who don't understand R&D as much, to just sort of simplify things as much as possible in terms of sort of the process with mice and validating the situation?
Yeah, no, I think Peter did a good job on that. It is a relatively quick look. I mean, we're by going directly into the brain on this, we'll be able to observe before we get into the more challenging aspects of developing a molecule that can be blood-brain barrier penetrant and work in humans. So again, it is just being cost efficient and cost effective, but also moving with urgency and speed to sequentially de-risk an opportunity. But to Aaron's earlier point, this and also we've done this with a couple other projects too, where we're looking for things that we think are higher probability cases. So in this case, we've seen with galactin-3 that it's been successful in other cases in humans. I am be sure of that, but I'll be corrected by Peter if I'm misstating. So if we can actually prove efficacy against Alzheimer's in the brain and neuroinflammation in particular around this overexpression in the microglia, and I think I touched on this in the Twitter Spaces we did. The microglia are kind of housekeepers in the brain, and they can be, they're normally very good for us in that they clean up the garbage in our brain and keep us healthy and cognitively fit. But when they get out of control, or they get tricked into thinking there's a problem in the brain, they do the opposite, and they start driving neuroinflammation and killing neurons. And so with galactin-3 being overexpressed in these microglia in Alzheimer's pathology, this is potentially very promising, where we've got something that we think can work in humans because it has, and now we can apply it to a different condition, which is in the brain, where we actually have very strong reason to believe it's playing a role in the disease category. I think this is great than something that's completely new, that's never, we are just trying for the first time. And so we've looked for a couple of these cases where we think we might have a better safety profile for a therapeutic, and then go after a new target that has to do with a brain disease, because then that kind of takes some risk out of the investment for us, and in some cases quite substantial amount of risk out of the investment, and that will help us in the pathway of getting it to clinical trials if successful, and give people more confidence in putting capital behind it earlier, like this. And then that the more that we're successful with those types of things, the more confidence we build in our ability to find and translate in other indications or other targets, and that just builds the confidence in our organization overall. So we think we like this project a lot for those reasons, and think there'll be some early signal that would give people comfort to continue to support this project at higher levels as we move forward.
Cool. Raphael and Lucas, want to give you guys the chance to chime in if you have any thoughts, whether on the science itself or on the sale, and also just kind of on the model that I guess we're validating with this new season of Bio. So any kind of initial thoughts that you guys have, or reactions to the sale? And while you're talking, I just want to make sure, are folks able to see my screen? I've been having a little trouble sharing, but just want to, can folks, is that showing up? You guys able to see my screen at all or no?
Just a black screen.
Yeah, just like
Okay. I've been trying to share the app just so people can get a sense of where to find more about. I'm not sure why it's not working. Let me give one more try. Oh, here we go. Got to add to stage. Okay, cool. All right, cool. So yeah, I just want to turn to, by the way, we're looking at the Bio app where you can see information on a Neuron Gale. If you go just to app.bio.xyz, go to our launch pad. This is the sale that just went live. So now we're finally live, and it looks like on the market's already done close to a 2x from the ignition sale. And just this is where to go for all information, how to get involved on the project itself. And yeah, so with that, any sort of initial thoughts from your end, Lucas or Raphael?
I can start really just quickly because I think something that it would be interesting to know as well from your side, Peter and Brian, it's on the IP landscape. Where do you see some possibilities? I think it would be interesting to know. Is it on the small molecule design to inhibit GAL3? Is it on the aspect of designing the brain-blood barrier penetrant? Where do you see the value once you prove mechanistically that this makes sense? I also think that there's signals that allow to move towards that direction. But in the long run, in the overarching vision of where this is going, where do you see value that can create IP or something that can create any innovation apart from what is already done, which is targeting galectin-3 not in the fibrosis but in the directly in the brain for neurodegenerative disorders. So just want to hear your opinion there.
Want me to take this, Brian?
Yeah, go ahead, Peter.
Yeah. Well, the strongest patents you can get, the strongest IP protection you get, and the highest value, of course, is the molecule itself. So composition of matter patents will be the most valuable to develop, and that's in principle what we're going for. The molecules that we're using initially are already patented or not suitable for human use, at least as a tool compound. So we're really going to go for composition of matter, focusing on a molecule that can go past the blood-brain barrier and has a high affinity for galectin-3 and has all the other characteristics that you would like to have, like drug-like properties. So that is the most obvious route to go for. There are additional routes which you can think of, which are less interesting, but could nevertheless create IP. You could have a way to actually help the molecule to get into the brain. So other projects that we're looking at in Cerebrum to see if there are mechanisms that you can help the blood-brain barrier to open and have the drugs in that are normally not passing. And then, of course, let's say down the road, you can think about specific use patents, but in specific indications. But I think the composition of matter is the most important one, and also the most likely route. Very crowded space. That is traditionally very big. So I'm very confident that we would be able to create IP there.
maybe I can take the next one. I think we have touched on AI in quite a few times during this discussion. I wanted to hear from the Cerebrum team specifically how you potentially see AI playing a role, maybe in this project, but also maybe give you the chance to, let's say, situate this project more into or in general frame Cerebrum, maybe also openly as in the context of AI, how you're thinking about that and which role it would play, maybe more as an outlook. Yeah, probably a Brian question, but
Yeah, I mean, I'll start with it, and Peter can pick up maybe particularly as it relates to this project, or how we've been using AI to date so far in our preclinical screening work, is we've had a good deal of success with AI models and silico screening with our Fission Bio project so far, which I think has been playing a role in why we've been able to move through milestones relatively quickly and cost effectively. Broadly speaking, we're in the process of putting together a AI agent, a bio agent, I guess you would call it, called Cerebre. So that should be coming soon to market from us. And Cerebre is an interesting one because we've got a partnership through a sister company with Xponetic, which I also run. And it's more on the front end, proactive, preventative brain health side, and we're working with the University of Texas at Dallas and their Center for Brain Health, because what they've done is they've created a battery of assessments that you can take that are non-invasive and relatively quick. You can do them in about 45 minutes or so. That they've coalesced into a score called the Brain Health Index, and then there are sub-factors to that. And what it and it's all been scientifically validated against fMRI data. So the outputs of the index are traced to actual activity in the brain. So that's why it's a non-invasive metric now. And so they've been implementing this over many years through tens of thousands of participants in a longitudinal study. And so we got really great data that shows, based on lifestyle behaviors and age demographic, age, gender, and such, what changes happen in brain health over time based on what you do in your daily life. And so we have it, and they also coach people. So we have an ability on the front end to actually look at brain and measure the brain in a diverse population of people over time. And so we can train up a model on that, which can act as kind of like a coach and an advisor for you on your brain health. And then on the flip side, as we look at our projects like Percepta and Fission, where we are using models, and Percepta, where we're running a decentralized trial, and we're actually using the Brain Health Index as a experimental endpoint on it, we can feed that data into the agent too. And then that starts training up an agent that's more on the exploratory research side. And so now you can start having more scientific interactions with the agent. That'll also be trained on scientific literature to try to find new opportunities for therapeutic development, but it'll also be tied to the agent that also can have a look at brain aging over time. So I think one of the things we've not had is something that when we're looking for therapeutic opportunities is to actually have a system that's been trained to look at how the brain changes over time across a diverse population of people, based on a lot of other interventions, and that'll be tied to wearable data too. So it'll be fairly comprehensive on a real world data basis, and we're going to be trying to incentivize collection and participation of that data. So that's where we're going with it. I really, this will get rolled out in phases, but I think an agent like that'll be fairly comprehensive and have a global population of people engaging in it on a more consumer basis, but with scientifically validated markers like the Brain Health Index, connected with training it up on science, and then feeding it more and more data from the projects like Gale and Percepta and Fission that we're doing. Gets very interesting over time. So it'll be early phases. I think it'll be a valuable consumer tool, And then I think as we kind of get into 2026, mid to late next year, I think it should start demonstrating some real promise on the research discovery side. But in the meantime, we're doing more engagement with AI and in silico screening in the preclinical side. So we're working with a couple interesting partners there that have been quite effective and cost effective. And so we've been learning on that side too, and I think we seek to apply that to almost every project that we start now, because we've seen such benefit from a couple of our early projects. I don't know, Peter, if you want to pick up on that part of it of what's actually actionable today, while we get Cerebrate launched and trained up over the coming months and year.
Yeah, very briefly, Brian. I see we're almost running out of time. The first most obvious one is where we have been looking for many decades, is to actually on the chemistry and target side to be more efficient rather than blunt screening. So the AI can really play an enormous important role in finding the molecules very quickly and very effectively, rather than having to synthesize them and try them out in screening. In layman's term, what we're looking for is in silico to find the perfect key that fits into the lock. And if you're thinking about these, I think it's called dimple keys, which have different holes drilled in them, a very sophisticated lock. It's like having the computer trying to have different holes into the key until it finally fits, without actually having to make the key. And that's where we already can deploy AI, and you can screen billions of compounds that have drug-like properties in silico. And then before actually doing a drug screen, synthesize only a few that are called your highest hits, and then very quickly go to those molecules that, technically spoken, have all the characteristics that you're looking for. Now, we're still in the early days. There are a number of companies that are active in this space. We're working with one. So that's the obvious thing to kind of use in the very early process. And then, as Brian was saying, as you go more into the complex matter of biology, you can use AI in various ways of trying to understand your data, looking at your data that you are generating, and have that analyze it, rather than having a human brain trying to understand the very complex data that we're looking at. So I could go into more detail, but I think that's how you're along the line. And also when you're thinking about, and that's what Brian was hinting to, if you're thinking about clinical development, where of course in this case we are still far away from, but it's good to think of in translational matter, is to how are you going to measure effectivity or efficacy? And so far we have been relying on a number of very subjective endpoints sporadically. And if you could combine the totality of what's going on in the brain and also your more, let's say, objective biomarker measures, if you could combine them and trying to understand what actually happens when you're starting to treat patients, you have a much better understanding of what's happening. And I think that's where the future lies also in the clinical space. It's not where it's unlocked at the moment. Most companies are actually unlocking the potential in the discovery space. But I see the future benefit actually in the clinical space. And I think that's where we're moving also with Cerebrum.
All right. With that, we are coming up on the hour. So I think we should wrap up things here. I want to thank everyone for joining, and also congratulate you guys on a pretty epic start to Neuron Gale. The sale, again, was over 14x oversubscribed. It is now live and trading on the open market. If you're holding Gale, you can stake it for what we're calling new token multipliers on your BioXP. Again, BioXP unlocks access to future sales, and we'll have future ignition sales starting next week. We're expecting 3 to 4. So the velocity of our launchpad is really going to start to ramp up after we've sort of validated it through this initial sale. So again, if you're holding Gale, feel free to stake it, or you can acquire it on app.bio.xyz and stake it as well to get those rewards. And then follow along Neuron Gale at Neuron Gale on X to follow along with the progress of the project, to see this galactin-3 inhibitor validated, hopefully inside a mouse model. And so super excited to see the science progress. And thank you guys so much for joining and sharing everything. All right, we will wrap here.
Excellent.
Thanks for having
you guys.
Take care.
Thank you.
Bye-bye.
Bye-bye.